Welcome back. I'm doing chapter 10 here for DNA structure. A lot of this is material that you covered in Bio 1. So I want you to go back to your Bio 1 notes or your bio notes from high school or whatever you have and look over your understanding of DNA. In micro, in other classes, you have definitely covered this again. We are going into a little bit more detail, maybe, but probably not than what you've covered in the past. So a lot of this may be review. I'm hoping it is. But then we're going to get into more detail than you've covered in other classes about the types of enzymes involved in copying and reading and maintaining the DNA. So hopefully this will be a little bit of review and very little new stuff for you. We're going to look at the central dogma here. And again, when we cover transcription and translation, we're going to look at some of the classic experiments about Griffith. We've already covered Griffith. We've already covered Avery, McLeod, and McCarty, Hershey, and Chase in other aspects of this class already because they fit in really well. So I'm hoping this will be the second time you've heard some of this, if not the fifth time, depending on how many classes you've taken. And we're going to start in this video looking at the structure of DNA, things like what are nucleotides versus nucleosides. In video 2, I'll continue on looking at experiments about the structure of DNA, and then we'll look at things like what does it mean to be 3 to 5 and 5 to 3, the shapes of the nitrogenous bases, what does it mean to be anti-parallel but complementary, and then we'll look at a little bit of RNA DNA structure, and finally the things I think might be new. We're going to look at ways that we understand DNA in a bioinformatics or a molecular analysis kind of structure. So how do we confirm this stuff that we've accepted all along for our whole lives, that DNA really looks the way we say it looks? With that, we'll get started with the idea of genetic material. We've been talking about genes since day one. That's genetics. It's kind of the class about genes. Genes are where you have your code inside of you, and it's how you pass your information on to the next generation. It is ultimately where we'll take it at the very end of class. It's where variability is. Natural selection requires variability to select on something. If you don't have any variability, you can't adapt. There can't be differences in the population. I want to start hinting at that because our last section is on population genetics and how we look at changes within a population, and we predict how maybe we need to develop new treatments or funding, and also how we understand evolution to happen in the real world. We're going to start looking at how genes play a role in that. In order to be genetic material, you have to be able to replicate, because you have to be able to pass on an accurate copy. It has to be information that can be stored. It has to be accessible in a way to allow that information to be expressed, and it has to be able to be varied. So over time, little changes can sort of randomly appear. There have to be mutations. So these four count as anything that fits those requirements can be genetic material. We'll keep coming back to this idea of the central dogma, and this has kind of fallen out of favor a little bit as we have moved into a more modern understanding of genetics. But I want to start here with this idea that the information of flow in a living organism is one directional. Information is stored in the DNA. That DNA storage of information is accessed via transcription. Transcription is where you have some molecules that make a copy of one region of your DNA. If you're a bacteria, that could be an entire operon or a polycistronic gene from last lecture. If you are a eukaryote, it's normally just one gene that may be all or part of a protein ultimately, but that transcript is in mRNA. It's a different kind of molecule. That mRNA or messenger RNA is then moved to a ribosome. The ribosome translates, it takes the information in the nucleic acids of the RNA and converts them into amino acids of a protein. That protein is then ultimately synthesized as a polypeptide. It's folded into a functional protein. So I took this from a more basic textbook. I think that this is, it's very similar to the one in your textbook, but I feel like your textbook is trying to show you a little bit more than we need right now. We'll come back and use those diagrams as we get past transcription and translation. Chromosomes have DNA, And we've talked about this all along. We know that the genetic material is on a chromosome because of work that Morgan and his cohort of researchers did. We know that DNA is actually a type of molecule called deoxyribonucleic acid, and it's made up of 4 basic nucleotides. Proteins, we also know, are important in expression of those genes. But when people were studying it for the first time, it looked like proteins were the more interesting and varied molecule. Proteins are made up of 20 versions of amino acids. They have 20 monomers. And those 20 monomers seem to, on first blush, have the ability to communicate more complexity. An alphabet made of 4 letters can communicate less than an alphabet made of 20 letters. And so the assumption was that proteins were what was actually holding the genetic code, and DNA on the chromosome was actually just structurally holding it together. As people argued back and forth, the consensus by the early 1900s was that the actual information on a chromosome was contained in the protein component, not the DNA component. And this really kind of sits on this idea that there are only 4 nucleotides. And they assumed that if it was structural, that when we looked at DNA, we would find that each nucleotide represented about 1/4 of the mass of the DNA. It's the tetranucleotide hypothesis. This has been rejected many times. No one thinks this is the truth anymore. But that tetranucleotide hypothesis was a structural component of DNA, helping it to hold the necessary information that was actually contained in the proteins, according to people in the 1920s and beyond, actually, the 1940s even. So we'll head back here to Griffith. We talked about Griffith when we talked about transformation. I want to highlight the same exact experiment that he did, where he had Streptococcus pneumoniae. This has changed names. It's Diplococcus now. I'm going to go back because when you look it up, you'll see it as Streptococcus. We call the bacteria itself has changed name, but it's not relevant to this, only if you're looking it up. The Griffith did an experiment where he injected rough bacteria and smooth bacteria into mice. When he injected the rough strain into a mouse, the mouse lived because the mouse's immune system can fight off the Diplococcus, and it doesn't kill the mouse. In this smooth strain, and at the time in 1928, they just knew that they were smooth. They were injected into mice, and the mice died. So we had not mouse-killing bacteria versus mouse-killing bacteria. So I don't know how well you remember this because we mentioned it in passing, but when Griffith denatured, he heat-killed the smooth strain and injected it into the mice. Do you remember what happened next? Did the mouse live or die? Oh, these mice were fine. Hold on. It turns out if you kill bacteria and all their insides fall out, mice live. Because it's dead bacteria. So dead bacteria and not mouse-killing bacteria don't kill mice, and mouse-killing bacteria kills mice. None of this is shocking. The next bit was, when Griffith took the rough strain and mixed it with the dead strain and injected it into the mouse, so not mouse killer, put it in a mouse, what happened to the mouse? Yeah, he died. Oops, he died. Which doesn't make sense. And we talked about this when we talked about horizontal gene transfer. Griffith described this process as transformation. He said that the rough were transformed by some substance inside the smooth strain, and that smooth strain took over and turned them into live, smooth strained mice, which is actually what is in these mice, these dead mice. You now know that what happened is these rough bacteria have a protein on the outside of them that, if they are competent, are allowed to take, they take DNA that is now in solution up and can express that as a gene as long as it has the full gene on it. They express the mouse killer, which is actually a protein coat that is coating the outside of the bacteria, preventing the mouse's immune system from fighting off the bacteria. We talked about transformation in horizontal gene transfer, but it was originally understood as a identifier of the fact that there literally was a code. Life is complicated, and it turns out there's a really complicated code behind it. Now, a little bit later, Avery, McLeod, and McCarty took the smooth strain bacteria and tried to see what component of them was actually the transformation factor. They took some S cells and they lysed them, meaning they popped them, and all their DNA fell out, right? But so did everything else. Their lipids, their proteins, their sugars, that's the polysaccharides, and the nucleic acids. And then they separated them with a centrifuge, which we will use soon, but you basically put the tubes in the thing, and it's a really elaborate spinning thing. If you've ever been on the spinning thing at the fair, you know what I mean. It like presses you against the wall. We separate things out by mass in a centrifuge, and then so they separated out all of the different biological molecules, and then they exposed them to rough bacteria to see which one transformed. Only DNA could cause transformation. They wrote a paper, published it, and were like, DNA is the transforming factor, and almost no one believed them. This is a very clear experiment with very few variables. It's very well controlled, but it just was so outrageous that most people assumed that their supplies were contaminated with protein, and so they hadn't properly separated the protein from the DNA, and so they were wrong. Later, this experiment was repeated much later, where it was definitely controlled for those molecules and shown 100% that DNA is the transforming factor. But it took one more experiment for people to accept the idea that this thing with 4 nucleotides was just as able to communicate information as something like protein with its 20 different amino acids or 20 different letters in the alphabet. We've also talked about this experiment, and this is the one that will really kind of tip the understanding of modern science into the idea that DNA is information. Hershey Chase, her experiment in 1952 centered around the idea of bacteriophages and E. coli. And we have seen this when we talked about infections in E. coli. We looked at T1 and T2 and T4 phages. She actually used the T2 phages and started out with an understanding of how phages worked basically. Basically, phages they're made out of a protein coat. They have protein on the outside and DNA on the inside. They are a DNA virus. When they infect bacteria, they bind to the bacteria and inject their information material into the bacteria and then take over the transcription, translation mechanisms, DNA replication mechanisms as well. And the bacteria starts making phages until it explodes. This is the lytic cycle. So they're reproducing baby phages. Don't call them that, but not really. But you know what I mean. They're making new phages. This is a process of transfection. We're asking what is actually the code being injected. And she asked whether was it protein or was it DNA. Well, DNA and proteins have different structures. They obviously have different functions. So she found atoms that were in one and not the other, and she radioactively tagged them. She used radioactive isotopes. In the DNA, she radioactively tagged the phosphate groups that sit on the end of each nucleotide. And in protein, she radioactively tagged sulfur disulfide bonds, which don't occur in DNA, only in protein. When she did that, she let the phages go and infect in 2 different samples. She had a sample where phages were radioactively phosphate tagged and a sample where they were radioactively sulfur tagged. The radioactively phosphate tagged ones were allowed to infect, And then she spun them in a centrifuge until all of the original phage protein coats fell off. And the only thing left inside was the transformative factor, the thing that was controlling the new reproduction of phages. She did the same with the sulfur sample. She radioactively tagged sulfur. She took the she took the infected bacteria with the phages attached and spun them out. The phage empty containers are shot off. She looks at the bacteria and what happens next, and only the transformative factor is included. The question becomes, which sample, DNA or protein, at the end has the radioactive material inside the E. coli? Well, she showed that the radioactive material was only found in E. coli where it was tagged with the DNA. She definitively showed that it was DNA and not protein. In her protein samples, all of the radioactive material got tossed off with the phages when they got centrifuged off, and none of it was left inside of the DNA. Now, this is super helpful considering how simple the phages are. You couldn't do this experiment with something like a COVID virus or a flu virus because they incorporate a lot of proteins in what they pass into into their host. But for a phage, they're fairly simple structurally and component-wise. I have a little bit of indirect evidence as well that I think you didn't cover in Bio 1, so I want to cover this as sort of we have this direct evidence through research, but we also can look at the effect of DNA and what it does and how it interacts in the real world and guess that it's important in coding. Gametes and DNA have the same amount of chromosomes and they correlate from organism to organism. So you and your parents and your grandparents and your descendants have the same amount of DNA in mass-wise in your gametes every generation. That suggests that that's important, that amount is important. It also, although the types of proteins vary in your body from cell to cell, your DNA does not. Inside of each cell you have with a nucleus, you have the same amount and exact full code of your DNA. When we mutate and we look at things that are commonly known as mutations, I'm down at the second part here, the UV light mutates the code, and that causes breaks and damages. So when UV light shines, and I don't know if you did this in micro or not, but did you do the experiment where you shined the light on the bacteria and you interfered with their reproduction ability? What happens is it causes the bonds inside of the DNA actually to break and form into twisted ways. It causes it, when it goes through replication, to replicate incorrectly. It mutates. UV light damages DNA at 260 nanometers. Proteins don't absorb 260 nanometers. They absorb 280 nanometers, which is not a damaging wavelength and is not known to have any mutagenic effects. So if I shine UV light at 280 nanometers, and that's the wavelength of the light, on you, it doesn't do anything. It does, even though it's damaging, affecting being absorbed by your proteins. If I shine UV light at 260 nanometers, it can cause mutations. That suggests it's the DNA, when it's damaged, that's leading to mutations. And I know you already believe me on this because we've kind of talked about it at the beginning. It's hard to separate the learning about DNA from our understanding today, but our learning about DNA is in modern times. People were driving cars and flying in airplanes before we understood what the code even was inside of you. This is a very new understanding. And that was with Dr. Flisser the other night, coming in and being like, we were laughing about, the fact that you can do what you're doing in the lab is just like 20 years ago, that was bonkers. It's imaginary. And now we can talk about it all the time. I want you to consider how fast our understanding of this has increased, just the ability to manipulate the molecules themselves into things like transgenic and recombinant DNA. Holy cow, we're doing that in our lab But 20 years ago, that was something that we would have watched a little movie about, and we might go on a tour and see some big technology corporation do it. But the idea that we can do it in a lab, absolutely, people have worked really hard in the last 20, 30 years to understand and how our code actually works. And it's really driven heavily by trying to understand how to cure people of diseases. We can look and link eukaryotic DNA segments to actual eukaryotic expression. We can tie this back to the idea of how do we look at the specific code in bacteria and then bring a eukaryotic gene and tie it to the bacteria and cause a bacteria to express a eukaryotic gene. Holy cow, this is what the pGlow experiment is all about. We are tying a jellyfish glow-in-the-dark gene into our bacteria, and hopefully in the plasmid that we insert, if it will uptake it, we will splice genes into two different organisms. We can do that by manipulating the DNA. It's not the proteins. We can directly do it now. The viral genome itself is not always DNA. Things like COVID, the flu, are actually code for RNA. This actually gives us some hint of the evolution of DNA as well. The RNA world hypothesis suggests that RNA evolved first. We think that because it tends to be simpler, shorter, and single-stranded. Not always, but it can be. When we look at a lot of viruses, things like the tobacco mosaic virus was the first one it was seen in, but even today, and this is a tobacco plant virus, we can demonstrate that RNA is in those cases used as the code. When I copied RNA and I'm using RNA as my genome, I'm a virus now, I use an enzyme called RNA replicase. Since we're here, and I think this is the first real enzyme we've talked about, when you see A's at the end, that tells you that this is an enzyme, a protein that is functioning to make a reaction happen faster or with a lower initial input of energy. There are some few, and we'll look at them, enzymes that are made of RNA. The fact that RNA can serve as a code and a functional molecule makes RNA a very versatile thing, but it's fragile. It's very susceptible to mutations and breakdowns, whereas DNA structure, as we'll see as we go on, is much more robust. Having those two strands wrapped together tightly all the time means it's less susceptible to damage from the outside, as well as it can hold on to the code a little bit better. We look at retroviruses, viruses that start by injecting their RNA, and then they have an enzyme inside of them called reverse transcriptase. That reverse transcriptase reads the RNA and reverse transcribes it into DNA. Some of the viruses we're going to talk about, and you talked about in other classes, are RNA retroviruses. I have some questions on the homework for this week, asking you about a few viruses. Read them carefully. Don't just assume because it's complicated or fast to replicate that it's a retrovirus. Read the instructions for each virus or the description for each virus very carefully. Reverse transcriptases are something we are also going to use in the lab. They allow me to do something like insert a piece of RNA in a vaccine and then get your body to make proteins from it, reversing the process of transcription. With that, I'm going to end there. I know that's really brief, but it's a lot of experiments to think about. The next part I'm going to talk about, I feel like here, the next part I'm going to talk a little bit about nucleotides and how they're built together. I think you know this. Get your old notes. Before you start video 2, sketch out how DNA looks. What does it mean that it has directions? What does it mean that it has a spin? What's holding the bonds in the center together versus the bonds on the side? Think about those things and then start video 2. You know everything except for the very last 3 slides. I'm pretty sure. And then I'll see you in the next video. All right. Bye-bye.